So it's 2 minutes past. Let's begin. Hello, my name is Isabel Mendoza. I'm the Chief Communications Officer with the Global Plant Council. Welcome to a new webinar in collaboration with the Plant Journal. Today we will be discussing receptor signaling, function dynamics, and evolution with 3 fantastic speakers. But first, a little bit about the logistics of this meeting while we are live here in Zoom. The chat is already working and it's working well, as I can see. You will be able to talk to us, the panelists and organizers, and with each other through that chat. Say hello, introduce yourselves, make comments, whatever you want, you are free to use it. But remember to send your questions using the Q&A feature in the bottom of your Zoom screens. There you can send the questions about questions from others, make follow-up comments if you wish, and it's possible that you can also see your questions answered in writing before we get to the Q&A. We will answer questions only at the end of the session, so please make sure to be clear about to whom is your question addressed. Finally, about the certificates of attendance, this is a new thing that we are doing. We will send them to your emails in the coming days or week. The only requisite is to stay connected for more than half of the talk's durations, which means usually, I assume, about 30 minutes. And as always, thank you for attending this Global Plant Council webinar series. And I will pass the turn to Katherine Denby. She's the editor-in-chief of the Plant Journal and the moderator for this session. So Katherine, welcome again, and the floor is yours.
Great, thanks, Isabel. And if you could put up my slide, that'd be great. So welcome everyone. This is now the 3rd Global Plant Council Plant Journal webinar, and which we've all had great turnouts, and again we've got some really exciting talks. I just wanted to give one slide on the Plant Journal for those of you that perhaps don't know the journal as well. So yeah, as Isabel said, I'm the editor-in-chief, and we publish a large number of plant science articles, over 600 last year, with more than 4 million downloads of those articles. We are partly owned by the Society for Experimental Biology. So for me, that's really important because it means that a proportion of the profits from the journal come back into science, into the society. So the society supports people with travel grants and organising meetings. And the Plant Journal, we also have a couple of schemes where we give Best Paper Awards each year. And we also support 2 early career group leaders, principal investigators each year, and then get started. And also as a society journal, we are very much about making it a positive author experience with our journal. So we train, and we have an editorial board who are very experienced and work together to try and make as fair and sort of transparent decisions as we can, and also timely decisions. So you can see last year that the average time to the first decision was 18 days. And we've also made the submission system very easy. So please send us your great science, and we look forward to interacting with you through the journal. However, now it's my pleasure to start the webinar series by introducing our first speaker, who's Johana Misas Villamil. Johana is a researcher in Professor Dolman's group, terrestrial microbiology group at the University of Cologne. And she also belongs to the CEPLAS Cluster of Excellence in Plant Science in Germany. Johana is interested in the interaction between plant and microbes outside plant cells in the apoplast, particularly dynamics between plant proteases and microbial protease inhibitors. And so she's going to be telling us about that today. So Isabel, if you could put up the slides. And just to remind everyone again, please put your questions in the Q&A function, and we'll be answering those after all the talks.
So, thank you very much. Thank you, Katherine. Thank you, Isabel. for this fantastic opportunity. I'm very happy to be here. And today I wanted to talk about CYP1 peptide signaling, biogenesis, function, and evolution. This is now a trip of more or less 15 years of work that I'm trying to summarize in the next 20 minutes. So let's go for it. So during the course of plant microbe interactions, plants have to deal also with pathogens, and this means that they can be infected. So good pathogens will suppress plant immunity, or they can also be resistant by inducing some immunoresponses. And we know actually that one of the important hormones against biotrophic pathogens is salicylic acid. So if pathogens are able to counterattack the salicylic acid system or inhibit salicylic acid responses, then the plant will be infected and they will cause disease, like in the case what you see here, Ustila gommatis infection, which is a biotrophic pathogen that infects maize. And this is our model system in the laboratory. On the other side, if pathogens are unable to counterattack the salicylic acid hormone-induced response, then the plant will become resistant, and then we will have, of course, happy corns and happy plants. So we also know that there are very important molecules also in the responsible of the salicylic acid pathways, and one of these proteins are the so-called papain system proteases, which are hubs in plant immunity. We know that salicylic acid itself activates these proteases, but we also know that somehow these proteases are able to also keep a loop in the salicylic acid response to maintain a feedback loop of activation. But it was not really clear how does it work. So for the first, what we used was a tool that we call activity-based protein profiling, in which we use a probe that specifically labels the active site of proteases, in that case of papain system proteases. And what you see here is that when we use total extracts of leaves that have been treated with MOC or salicylic acid, you can see that the salicylic acid treatment shows bands that are actually more abundant. So you see more signals in there. This already indicates that we have an activation of these enzymes in the salicylic acid treatment. So when we do this, also we test the apoplas. Also, Katherine mentioned at the beginning, we are very interested in the apoplas because it's the first interface where actually plant proteins and microbial proteins interact. So we also test the apoplas. We could find actually 6 or 5 apoplastic cysteine proteases that are also active in there. And you can also see here that when we I need to move my, sorry, now. When we inactivate these proteases by using an inhibitor that is so-called CC9, there is endogenous inhibitor, or a chemical inhibitor like E64, we are able to avoid the salicylic acid response that we read in this case for the immunoresponse by the PR gene expression of pathogenesis related 1 expression gene. So this also shows that PLCPs are actually required for the immunoactivation. Oh, sorry, do you see my slides?
It's still the PLCP one.
Yes.
I see. I pressed something and I cannot come back to my slide now. Let me see how I do this now.
You could always reshare if you want to.
Yeah, okay, I think I'm back. Sorry. So, well, PLCPs are required for the immunoactivation. So the question now is here, okay, if we have proteins or proteases that cleave peptides, so maybe there are peptides are actually involved in this activation. So let's hunt for some peptides during the salicylic acid response. And this is what we did here. So we did treatments of salicylic acid and MOC. We did an extraction of leaf apoplastic fluids. We then separate this apoplastic fluid to the high molecular weight and low molecular weight fractions, meaning the peptide fraction, and then we analyzed the peptide fractions by mass spectrometry, making a native peptidomics. So only peptides that are produced during this response have been then subjected to or have been identified during mass spec analysis. We were interested in identifying peptides that were specifically found in the salicylic acid response and not in the mock. At that time, we were very lucky. We could find actually only 4 peptides that you can see here, and they were called the candidate peptides. What we did was we synthesized these peptides then again, and then tested if these peptides can then produce a response in plants, and this is exactly what we did. And we can see that the PR gene expression of PR responses are actually upregulated, specifically one of these peptides, which is the so-called candidate peptide 2. In that case, we call it now CYP1 for ceamide immunopeptide 1. So where does this peptide come from was actually the next question. So we can see that before that, actually, that this peptide itself, when we again infiltrate this peptide in the plant, can activate again papain-like system proteases, as we know before from the very beginning of the introduction, similarly to the salicylic acid response. So if this peptide can then activate salicylic acid responses, then we assume that this peptide will be then detrimental during infection. So this is what we did. We use our system, Ustillagomaides. The SG200 is like the solo pathogenic strain, for us today the wild type strain. And then we did also a mutant that produces this peptide also during infection. And I hope that you can very well appreciate that the SG200 wild type causes tumors in the leaves, whereas these kind of tumors are also lost during the production of the CYP1 peptide. That means that CYP1 restricts Ustillagomaides infection, but also CYP1 induces salicylic acid signaling. And this gives us also the coming back feedback loop. Actually, CYP1 is the response that from PLCPs will be produced and again go the feedback loop to the salicylic acid signal. This is what we discussed before. So now, where does this peptide come from? Well, we have a very small protein here of around 137 amino acids. Then you see here in red our peptide. This is the CYP1 peptide. This protein actually is kind of unknown, doesn't have any motif, doesn't have any signal peptide. But what you can see is a very high load of some amino acids, like for example arginines, and this might play a role later on during the presentation. I will show you later on. So what we checked first is where is this protein localized. So we first did some localizations in Nicotiana ventamiana, but also in protoplasm. And for the first localization, it looks like it would be kind of nuclear localized, but also some cytoplasmic localization could be. Then we test specifically in the protoplasm, because this nuclear localization could also suggest actually that we have some ER localization. This is also very clear. So what you see here is actually we use an ER localization signal that is so-called the KDIL signal. Then we overexpress our PROSIF with GFP, and in the merged picture you can see that we have a quite good colocalization to the ER signal, but also very strong signal in the nucleoli, which is very well known for stress proteins that tend to have a load of arginines in there. So when we look at these arginines, actually we see that they are also in pairs. So we thought like these arginines might be important signals necessary or maybe require also for the localization. And indeed, when we do mutations of these arginines, you can see here in the mutant that we lose this nucleoli signal, actually, which is the kind of the stress response that we are supposed to be. And then we thought like also this signal might be important for the recruitment of membrane trafficking platforms. And this is maybe how this peptide or this protein or part of this protein can go outside the cell, because as mentioned before, this doesn't contain a signal peptide, and CYP1 was found in the apoplast. So to continue this story, we did some more biochemical analysis in which we did some fractionations in overexpressed samples in Nicotiana ventamiana system. And then what you can again appreciate here is that we have in the different fractions, specifically PROSIP label in the microsomal fraction, which is actually the one that might be correlated to the ER association, but we have also the cytosolic signal. But you can also appreciate that we have maybe 2 to 3 bands, mostly in this gel. These bands might correlate to first a full-length protein that you see in the first place, and then you have some cleavage products that we could recognize as a part of the N-terminal that is cleaved, so basically it's only the C-terminal portion, what we can see here, also some GFP alone, and there are some signals that don't contain the GFP that we cannot observe in the GFP gels, but also we are not observed in the CYP1 gel. This is a blood. This is a blood specific for the CYP1 immunopeptide, because as you can see here, these fragments might be too small to see on gel. To be able to recognize these fragments, we decide to make an ELISA to monitor the release of the CYP1 peptides. So, and as is very well known for animal cells, we have a very specific antibody. We use the CYP1 antibody we use to coat the plates and to identify small fragments that contain CYP1 peptides. For this, we have two controls, of course, our wild-type protein containing the CYP1 peptide and the pro-CYP1 mutant that contains these red lines here. This, if you remember, are the arginines that we saw before. So we changed these arginine duplets or alone arginines for alanines. So we assume that this might impair the release of CYP. That was the first assumption we had. And when we tested in the ELISA, well, you can here see that there is a tendency for the mutant to accumulate or to retain in the microsomal fraction. It's exactly as we see also before in the microscopy. And there is a tendency also for the wild type to be more in the apoplas. In the tendency I mentioned just because this is not significant, but when we test the samples in the apoplastic fluid, we can see in the high and low molecular weight fraction that there are more peptides coming out from the wild type molecule than from the mutant molecule retained in the apoplas. So this suggests that there is an N-terminal processing that enables CYP1 translocation to the apoplas. We did then mass spec analysis of these samples actually, and then we, with a pull down of GFP pull down, we could identify mostly peptides generated at the C-terminal part of the protein. So you can see here in the upper part, N-terminal, lower part, C-terminal part, and you can see here the peptides that we identify. We also identified the CYP1 peptide in these fragments. So for this now on, we call the C-terminal part CT-ProCYP, for the C-terminal pro-CYP1 that we think is the protein that is generated. So for now, what we have is actually an ER association, and we have also an N-terminal cleavage that I can show you before, and we think there is some enzymes that might be internally cleaving then this protein to release the C-terminal part that should be going to the apoplas. So which protease mediates pro-CYP N-terminal cleavage? We know that should be arginine dependent, intracellular, and stress related. And we when we think about all these three characteristics, we come up with metacaspases. So we first check which metacaspases are at the same time expressed with the pro-CYP gene, and we find actually only one metacaspase. This is the MC9, is a type 2 metacaspase. And when we recombinantly produce a metacaspase and pro-CYP, we can see actually that there is and combine these two, we can see that there is cleavage with a higher concentration of the metacaspase of our pro-CYP, and this cleavage can be abolished, of course, in the metacaspase mutant that is not active. Metacaspase we tested, indeed, it has an arginine cleavage site, so it's specific for arginines. And when we combine again these two samples and do N-terminomics analysis, and then N-terminomics, we can see actually which fragments are generated after the incubation of these two enzymes. We can see here in these lolly graphs actually that the new N-termini, which are labeled here in black, are mostly also produced after these arginine sequences, suggesting that actually these metacaspases again recognize the arginine sequences. And more interesting, if you see here this part between 85 and 105, this is the part that contains our CYP1 peptide. And if we do docking modeling using AlphaFold analysis, we can see actually that the pro-CYP1 part really binds or really kind of correlates or dock very nice into the active site of the metacaspase. And then you see this arginine 84 here, which is probably the arginine that is cleaved to release the CT-ProCYP also that is then found in the apoplas. But we can also directly see probably a release of the CYP1 peptide itself. We also tested in that case if the CT-PROSIP might be already a bioactive molecule using the Trojan horse experiment with Ustilagomyces. And what you see here is indeed the CT-PROSIP shows exactly the same phenotype of the CEP1 peptide, because we lose infection virulence actually of the Ustilagomyces in the presence of CEP1 of CT-PROSIP, whereas not with the SG200, which is our wild type. But when we mutate the CEP1 portion, only the CEP1 portion of the CT-PROSIP, we again return the wild type phenotype, meaning actually the CEP1 itself is the molecule responsible of the salicylic acid response, in that case of the virulence of Ustilagomyces. So now we have found a molecule that is very important in salicylic acid, and we thought, okay, how conserved is this now? So we went now to look for PROSIP genes, and that was when Corona started. This is again our Corona project, so we do some bioinformatics in here. And the first blast only went to CMIs, and we thought like, okay, this cannot be. So with some more analysis, we went to the Andropogonea family, and we could see actually that also in some of the relative species like Trypsacum, also this gene is not present. So PROSIP1 homologs are exclusively found in SEA species. So, and when we go deeper into where PROSIP is localized, we can see that PROSIP1 resides in a long terminal repeat in a retrotransposon in different chromosomes. So we have in chromosome 7, chromosome 8, and 2 variants in chromosome 10 with different rearrangements, all containing PROSIP genes. Some of these genes are also pseudogenes that we can see actually here in this table on the right. These tables are the so-called NAM lines that you can also see containing one or different copies in different parts of the genes. And we have also 2 lines like the Chi21 line that does not contain any gene at all, but also the P39 line that also has one pseudogene that doesn't encode for a protein. So doing some more analysis of the retrotransposon, we wanted to see actually how this retrotransposon is conserved in different or across the different SEA species. For this, we did an identification of the homologs LTR from Andropogonea again. We did a selection of representative copies among nearly identical ones. We started with around 60% homology, and then we did a phylogenetic reconstruction. When we do 60% homology, we end up again only with CMIS species, but when we go a little bit down to 54%, then we have some homologous in the Trypsacum clade, and these are the ones that we use in this phylogenetic tree as an outgroup. And you can see actually that we have again PROSIP LTR and this PROSIP pseudogene very, very correlated at the top as very similar sequences. So we think the PROSIP emerged through gradual mutagenesis because when we do an ancestral analysis of these sequences, you can appreciate in here actually the sequence conservation. This is only the part of the PROSIP, but actually we did the whole analysis with the LTR that you see here down there. And you can see actually that we have some insertion, deletions, and substitutions over time that might be responsible for the fixation of this gene and the genome of maize. We also think actually that this is happening probably due to some positive selection. Maybe that was already used for the salicylic acid response. So what is the difference in between the different chromosomes? For this, we use the different lines that contain the different chromosomes, did again some salicylic acid treatments, and we use the 2 lines that are our control lines, one that doesn't contain PROSIP at all, one that only has the pseudogene, and then after salicylic acid treatments, you can appreciate here with the red pluses that only in the green line, these contains actually only PROSIP sequences, or only PROSIP because we have a gene on the other side. The green lines with the red circles here are actually the signals coming from PROSIP coming from chromosome 7, but not chromosome 8 or chromosome 9. So it looks like chromosome 7 has somehow features that are upregulated after salicylic acid treatment. So we went to check to the promoter region, and what we really see in there is actually that we have 2 W boxes in this region. So you can see here the clade of chromosome 7, chromosome 9 and 10, and chromosome 8. And you see here on the right side that there is a position where the W box is conserved in all chromosomes, but on the left side we have a position that only appears in chromosome 7, but is not in the other parts of the chromosome. So we assume that we have a cooperative regulation with a worky protein binding after the salicylic acid response. So how to test this? We basically generate mutants of these regions when we use the wild-type chromosome, then mutated chromosome 7 and the chromosome 10 use a luciferase assay, and this is a very preliminary data. What you see here is actually that indeed when we do the salicylic acid treatment in this sample, we have an activation of this chromosome, but is of this chromosome signal basically, but it's not in the chromosome 7 that is mutated or a chromosome 10. That suggests that mutations in the chromosome 7 W box likely motive abolishes assay responsiveness. And with this, I want to go to the take-home messages. So we show you today, or I show you today, that the CYP1 peptide activates salicylic acid-triggered immunity in maize. The CYP1 is released from the endogenous propeptide ProCYP1 by a dual mechanism. First, we have metacaspase that cleaves the N-terminal part, and then we have a non-conventional secretion into the apoplasm. I also show you today that ProCYP1 is specific for SEA and evolved de novo from a retrotransposon. And the very last preliminary data that chromosome 7 might contain a W box that regulates salicylic acid responsiveness through a cooperative regulation. So thank you very much for your attention. I want to test the terrestrial microbiology lab, our sponsors, and the majority of the work that I have shown today, especially of the mechanism of processing, has been done by Maurice, who is here in red, and also all bioinformatic analysis have been now wrapping up by Yuki. And thank you for your attention.
Yeah, thank you, Johana. Yeah, Johana, that's really great. Not often you get to work on a protein that's so specific to one genus like that. So remember, please put your questions in the question and answer box. And one thing I forgot to say earlier is that the topic of this webinar, receptor and ligand interactions, is actually the topic of a special issue that will be coming out in Plant Journal later this year. And all our speakers are authors on papers in that special issue. So look out for those publications. Okay, so our next speaker is Satoshi Fujita from the University of Toulouse. And Satoshi also has a position in the CNRS, in the Laboratoire de Recherche en Sciences Végétales. And he looks at how hormone signaling and cytoskeletal organization is controlled or coordinated to control growth and development. So over to you, Satoshi. You're still on mute if you're, yeah. All right, yeah, getting your. You need to unmute.
Satoshi, you are on mute.
Great.
Can you hear me? Sorry. Can you hear me now?
Yes, we can hear you.
Okay, thank you very much. Thank you very much for nice introduction, and thank you very much for. I'm really happy to be here to show our recent results about brassinosteroid signaling and cortical microtubule regulation. Okay, first of all, I would like to highlight today's work. It's mostly done by Charlotte, previous PhD student. She's now working in University of Bellevue. And today's work is also our teamwork with a nice collaboration between in the team. So as you know, the plant from seed to seed, plants need to sense many stress, or they also need to communicate each other between the cells to have a proper development. During this process, they need to sense the external situation, biotic, abiotic stress, or communication, short distance communication or long distance communication is quite important. One of the major sensor in plants is a receptor kinase, as you know. Plants sense something on the extracellular domain and emit signal from the internal kinase domain. And many receptor kinases are assigned to quite a lot of events, including stress response and many developmental process and reproductive process. Among them, we use BRY1 as a model to understand receptor regulation and its downstream, because BRY1 is well characterized genetically, cell biologically, biochemically, or many aspects. This picture already shows how BRY1 is important. In the BRY1 mutant, the cell growth, plant growth is quite perturbed, and BRY1 is because this is a major brassinosteroid receptor, and not limited to cell growth or proliferation. They also involved in the differentiation, such as stomata or stem development. For example, when brassinosteroid signal is perturbed, abnormal amount of stomata appeared on the leaves. This also involved, BRY1 is also involved in stress response, such as heat or salt stress. For example, when plants are treated with BRY1, brassinosteroid, one of the potent brassinosteroid, the recovery rate of the plants is much higher compared to non-treated plants. At the molecular level, BRY1 perceived brassinosteroid together with BAC1 co-receptor and then emit signal. And at the cellular level, this most part of the BRY1 localized on the plasma membrane, and some parts of the signal we can see in the endosomal compartment. And in such context, our goal is understanding the downstream and regulation of brassinosteroid signaling. To do so, we conducted 2 hybrid screens together with cytoplasmic kinase domain as bait. And then we obtained well-known BRY1 interactor such as MAKR1 or SAC1 co-receptor. In addition to that, we also identified novel interactors. Now we call this as microtubule BRY1 associated proteins, MBAPs. Today's presentation, I would like to explain you why we call this protein as MBAP. In Arabidopsis genome, there are 3 putative homologs, and then we isolated all of them in our 2 hybrid screen. This region were isolated in the 2 hybrid. And unfortunately, there are no characterized domains, but among them, N-terminus region were relatively well conserved. And then this protein is found widely in the land plant. First of all, we check the expression region of MBAPs to ask whether BRY1 can function together with MBAPs or not. BRY1 expressed over the plant, over the seedlings, including the root tip and the hypocotyl. And MBAPs shows a quite similar expression. Of course, there's some difference in the intensity between the organs or tissues, but it also explains in the hypocotyl and the root tips, suggesting BRY1 and MBAPs can work together. We also tested the interaction between full length of MBAP 1, 2, 3 and BRY1 And in two hybrids, we could see the interaction all together of BRY1. And we also tested co-immunoprecipitation assay after expressing in tobacco. And we could co-immunoprecipitated BRY1 and MBAP. This was not seen from only expressing the M-cyclin. And then this interaction was not strongly affected by brassinosteroid treatment. And we isolated MBAPs as new interactor of BRY1. And we tested what is the function of MBAPs during the brassinosteroid response. For that, we used hypocutaneous length as proxy. As most of you know, brassinosteroid promotes the hypocutaneous elongation in wild type. Yeah, you can see after 100 nanomolar, 500 nanomolar, hypocutaneous its length is much longer than non-treated one. We tested single mutant, but single mutant shows a quite similar phenotype to the wild type. Then we tested double mutant, and then in double mutant, we could see the MBAP double mutants are more sensitive to brassinosteroid compared to wild type. And the other way around, for the sensitivity to biosynthetic inhibitor, it's less in the mutant. And we also try to complement the mutant. In this case, we introduced MBAP1 in double mutant of 2-3. And then after transforming this, the response recovered to the wild type level, both in brassinosteroid treatment and PPG treatment. These results indicate genetically MBAPs are negative regulators of brassinosteroid-induced hypocutaneous growth. And to pursue the molecular mechanism, we checked the canonical brassinosteroid signaling. In most of the case, brassinosteroid response is explained by transcriptional pathway. And in such pathways, BES1 or BCR transcription factor phosphorylation status is quite important. In the absence of brassinosteroid, BES1 is phosphorylated, and then this form is inactive. And after perceiving brassinosteroid, BES1 is dephosphorylated and gets active. This can be confirmed on the Western blot. When no brassinosteroid is treated, almost 1-to-1 ratio between phospho-BES1 and BES1. But after treating with brassinosteroid, non-phosphorylated forms start to accumulate. And we tested each double mutant, and it shows a similar response like wild type to the brassinosteroid, indicating MBAPs do not affect phospho-BES1 ratio. To be sure, we also checked the transcriptional status of BES1 target gene, BES1 direct target gene. We picked up two genes. One is upregulated, and the other one is downregulated. And in both cases, we could see the similar response to wild type. So we confirmed the MBAPs are not involved in canonical BRY1 BES1 signaling transcriptional pathway. And so what is the function of MBAP? To reveal that, we try to characterize MBAPs more deeply. First, we did BIFC assays, and here's negative control, and here's positive control, as BRY1 shows makes a homodimer. And the signal also reconstituted when we express BRI1 and MBAP. And then what was interesting for us is when we change the focus a bit, and then we saw the surface of the cells, and then only in this combination we started to see some filamentous structures, which was not seen in this control. So we assumed BRI1 and MBAPs may interact on filamentous structures. Then we checked the localization of MBAP in hypocotyl. We used native promoter here in triple mutant. This construct could complement the mutant as well. And we saw some filamentous pattern, but with fragmented. And it was not clear, but when we accumulate the signal from 200 frames at the same focus, we clearly started to see the typical cortical microtubule patterns. And this was also confirmed when we expressed microtubule markers. The signal well overlapped with microtubules. Then we wondered what is the function of MBAP on microtubule. So we crossed the mutant with a microtubule marker GFP map4. And in wild type, we could see the well-organized microtubule patterns in each cell. But in the double mutant of MBAP, we saw the quite random network of microtubules. This was also proved by quantification of isotropy. So MBAPs are required for proper microtubule organization. Next, we asked, what is the function against the stability? To do so, we add orizarin. We add orizarin, which is microtubule destabilizing drug. We observed microtubule over 20 minutes. As you can see, in wild type, the number of microtubule gets less and less, and which also happened in the mutant, but the response to the orizarin is more pronounced in the mutant. And this data indicates MBAPs stabilize cortical microtubules, or it's important or promote polymerization of microtubules. And then lastly, we asked, what is the link from BRI1 to microtubules through MBAPs? To confirm that, we checked the reorientation of microtubule, which is induced by brassinosteroid treatment. As you can see, in wild type, after adding brassinosteroids, the microtubule orientation angle of microtubule becomes from perpendicular to horizontal. This also happened in MBAP23 double mutant, and we quantified this, and then we found in the MBAP23 mutant, the response is more enhanced, and the speed is also higher. So we now assume MBAPs, the molecular function of MBAP against microtubule is MBAPs suppressing the BR-induced microtubule reorientation. To sum up, we found MBAPs as a new interactor of BRI1, and which localize to the cortical microtubules. And most of the elongation of the cells is induced by brassinosteroid is explained by a transcriptional pathway. but MBAPs are not involved in this canonical transcription pathway. And because of the reorientation assay, we think MBAPs suppress this reorientation from the brassinosteroids treatment, like this, from perpendicular to horizontal. Then this might be important for the suppressing cell growth because microtubule direction is quite important for cell growth. From here, I would like to show some perspective. As someone can imagine, some population of BRY1 could be anchored to microtubules through MBAPs, so which may affect the dynamics of BRY1. So I construct BRY1 MUS, and then to chase the single molecular dynamics of BRY1, we can see like this. Most of the signal stays on the same place, and then we chase the BRY1 signaling like this over 3 minutes, and then we could see the diffusion rate like this. Now we are testing the orizoline or brassinosteroid treatment, or also in MBAP mutants. And then another question from this research is usually microtubule orientation and cell growth pattern is well linked, but in the mutant, microtubule is random, but the cell looks elongated in the normal way, and then also the hypothesis itself is also quite healthy. So this means in MBAP mutant, the feedback loop between cortical microtubule and cellular elongation, this physical feedback loop might be perturbed. So we are now setting up some ablation assays or some mechanical assay to reveal this kind of phenomena. This is the last slide. I'm now interested in the role of microtubules. When we open the textbook, most of the textbooks just says cortical microtubules are important for guide rail for cellulose synthesis. And then recent research also shows tension sensor. And some research also shows limiting the microdomain is another function of cortical microtubules. And I also want to ask whether cortical microtubule have some relationship with plasma membrane protein, because microtubule covers a quite significant amount of surface of plasma membrane, but we still don't know the relationship with plasma membrane protein and microtubule except cellulose synthesis. So our MBAP research might have some open a new avenue to reveal the relationship of plasma membrane protein and microtubules. And thank you for listening, and I'm happy to have questions later.
Great, thank you, Satoshi. That was also a really interesting story. I'm sure there'll be lots of questions around that. Great, so now it's a pleasure to introduce our final speaker, Jacob Merlang. So Jacob's a postdoctoral researcher at UC Davis in the Schabec lab, and he's also working on plant hormones. Earlier in his career, Jacob has been working on linking mechanical stress to sort of electrical and calcium signaling, and now he's looking at hormone receptor signaling pathways that integrate environmental cues to control plant responses. So over to you, Jacob. Thank you.
All righty, thank you so much. Yes, my name is Jacob Merlang. I am at the University of California, Davis in Nitzan Schebek's lab. And in the Schebec lab, we are interested in leveraging structure function biology to study plant signaling and hormone mechanisms. So I'm going to show you guys some unpublished data today. So I'm excited to give you guys a little preview of what we hope to put out in the next couple of weeks. So kerykinin-sensitive 2, or Ki2, is a highly conserved plant hormone. It was originally identified as a receptor for fire-derived butanolide compounds, such as kerykins you can see down here. Now, what's interesting is that many plants, if not most plant species, are actually sensitive to these fire-derived molecules, which is unusual for plants that don't exist in fire-prone landscapes. And that is because, as I mentioned, Ki2 is an ancient hormone receptor. And you can actually see it arises evolutionarily pre-land colonization in plants, which is quite remarkable. Additionally, Ki2 has undergone gene duplication and neofunctionalization, and one of these events actually led to its sister receptor, D14, which is important in strogalactone signaling. Now, Ki2 is a dual enzyme receptor, and you can see in this video on the left of its structure that it's an alpha-beta hydrolase, and it contains a catalytic triad with a serine, histidine, and aspartic acid. And these are important in hydrolyzing pocket-bound ligands. Now, synthetic ligands such as enantiomeric GR24 that we see down here, as well as natural ligands such as kerykins, bind to Ki2, but the endogenous Ki2 ligand, termed KL by the field, still remains a mystery. Now, broadly speaking, Ki2 operates in a larger signaling module, and it involves the E3 ligase more accelerated growth 2, or MAX2, as well as the suppressor of MAX2-1, SMAX1. Now, SMAX1 and its homolog SMXL are important transcriptional repressors that repress Ki2-mediated downstream transcriptional activity. Now, Ki2 will bind to KL, and this causes a hydrolysis and a conformational change in Ki2, which subsequently results in MAX2 Ki2 complexing and recruitment of the SMAX1 proteins. The SMAX1 proteins are then ubiquitinated and subsequently degraded via the 26S proteasome, and the transcriptional brake is then released on Ki2-mediated signaling. Importantly, Ki2 also undergoes some proteolytic degradation, albeit at a much, much slower rate. Now, Ki2 is involved in a host of different processes, and we actually highlighted this in a review recently in the Plant Journal that you can scan the QR code here for. And it's interesting that Ki2 is involved in many processes in plants, including arbuscular mycorrhizal formation. It's important in abiotic stress resistance, including drought stress and heat stress. It's been proven important for germination, and more recently, actually very important for basal bryophytic plants and asexual reproduction. We heard a little bit about salicylic acid, and indeed, Ki2 also participate in some immune signaling, as well as its more canonical phenotypes, such as impacting hyphocaudal growth and root hair growth under phosphate stress. Now, this next slide is very busy, and I don't want to go through each of the hormones here, but I want to highlight that Ki2 is upstream and regulates a diverse set of hormone signals. But there are some fundamental questions that are still remaining. And that includes, for example, is all of the interaction we see in Ki2-mediated signaling through the transcriptional repression via the SMAX1, SMAXL2 module? Are there post-translational modifications that occur to Ki2? Is there an enzyme or a carrier molecule that potentially could hand off the KL enzyme, or sorry, metabolite to Ki2? And additionally, are there additional regulators or protein interactions occurring during the E3 module with MAX2, SMAX1, and Ki2? In order to get to this, we leveraged proximity labeling to investigate novel Ki2 interactors. And so we have Turbo ID here, which is a promiscuous biotinylase, which with sufficient biotin supplied will biotinylate or tag any neighboring protein within 10 nanometers. So if we fuse it to Ki2, the idea is that it should be able to biotinylate and tag any proximate proteins to Ki2. And if we have a control M citrine, we can develop an enriched proteomic data set showing for Ki2 interactors. Now, we have in our lab, we've successfully leveraged Turbo ID in a couple papers. And most recently, you can see here, my co-author, Daniela Rodriguez and I, we published a paper using abiotic stress and studying the enrichment profiles of E3 ligases. So it's very interesting. But back to the Chi2 story. So we have our stably transformed Arabidopsis plants with our Chi2 turbo-ID or M-citrine turbo-ID constructs. We can infiltrate biotin into those plants. We incubate them for about 60 minutes. Then we extract the tissue harvest and then protein extraction, followed by then purification and enrichment on streptavidin beads. We have collaborators at Iowa State in the form of Justin Wally, and his lab has been helping us generate the proteomic data sets where we actually see the enriched peptide differences. Now, the Chi2 peroxytome revealed interesting new interactors and pathways. And so I just want to highlight a few Go terms that we saw here. For example, we saw a response to keratin, we saw root hair initiation, and we also saw a response to abiotic stimulus. Now, these are all phenotypes that I previously mentioned Chi2 is important in. And so let's dig a little deeper into the individual proteins that we pulled out. Now, I pulled out many fascinating interactors in this data set, but I want to highlight just a few here. One is that we see indeed our positive control SMAX1 appears in our data set, showing that we do actually pull out known Chi2 interactors, which is very important. And then I want to highlight these two other interactors that we're going to talk about for the remainder of the talk. The first one is PAL1, and the other is MLP. Both these proteins are important in cinnamic acid signaling, and we'll get to PAL1 in a little bit, but I want to now move to MLP. MLP is an interesting protein in that it's a part of a larger family of proteins that are important in, as carriers of hydrophobic molecules in plants. And so we first wanted to validate that MLP indeed interacts with Chi2. So using a co-IP experiment in tobacco, we co-infiltrated our control citrine or our MLP target, and then, bait, and then we co-expressed Chi2. When we pull down for citrine or MLP, we only observe Chi2 detected in MLP co-infiltration plants, indicating that these likely interact. Secondly, we used a split luciferase assay where we have the luciferase protein into two fragments, the N-terminal fragment and the C-terminal fragment. And when expressed with two proteins that are in close proximity to each other or interact, we get the reconstitution of luciferase and luminescence, which we see here when we have Chi2 and MLP co-infiltrate in the same tobacco plant. Now, I mentioned that Chi2 is a carrier protein. So we recombinantly purified MLP, and we crystallized MLP and actually solved the structure. And what we find is that MLP has a big pocket that fits a nice hydrophobic molecule. And in fact, we're able to resolve the crystal structure with a benzoic acid fixed in the pocket of MLP, highlighting that, again, MLP may be able to carry proteins, carry small metabolites. This led us to question maybe whether Chi2 and MLP somehow interact in a way to regulate metabolite abundance, or somehow if MLP could protect Chi2 from metabolite dependent degradation. And what we actually saw was when we used a ratiometric reporter line developed previously in Dave Nelson's lab, where Chi2 is fused to an M scarlet, where we have a self-cleaving peptide sequence, and then we have a reference fluorophore in the form of Venus. MLP results in the increased stability and accumulation of Chi2. Now, we unexpectedly did not see Chi2 degrade when we added the ligand, as has previously been reported, but this could just be other reasons that we're not sure of. Now, MLP, as I mentioned, was involved in cinnamic acid signaling and had been previously shown about 15 years ago to be involved there. So we wanted to go back and make sure that that data held true. And in fact, MLP is transcriptionally sensitive to cinnamic acid. And you can see that here in the mock versus cinnamic acid grown Columbia 0 wild type seedlings. However, we did not observe any transcriptional differences when we tested the transcriptional abundance of MLP in Chi2 mutants grown on mock versus cinnamic acid containing media. Now, I mentioned that cinnamic acid is an important molecule, and it is in part very important because it's the starting point of the phenylpropanoid pathway. And the way that cinnamic acid is produced is from the deamination of phenylalanine via the phenylalanine ammonium lyases, or PALs. Now, remember, we saw PAL1 in our data set earlier come up in the TurboID data set. Cinnamic acid is reported to be quite biologically active, and in fact has been shown to increase lateral roots, inhibit hypocaudal growth, and affect changes in root hair characteristics. But I think for most people, cinnamic acid is best known for being the precursor molecule for all anthocyanins, flavonoids, lignin, and other important phenolic compounds in plants. Given this connection with MLP, PAL, cinnamic acid, and Chi2, we next decided to test how cinnamic acid impacts some of the phenotypes that Chi2 is known to exhibit. So one of the canonical Chi2 phenotypes is this elongated hypocaudal. And you can see that under mock conditions, indeed, Chi2 does have a longer hypocaudal. When we then grow the plants on cinnamic acid containing media, you see that there's a strong reduction in wild type hypocaudal length. Likewise, there's a reduction in Chi2 hypocaudal length, though it is still longer than wild type. Given that we are interested in understanding relative hyper or hyposensitivity to cinnamic acid, we standardized these experiments to look at the relative decrease from mock. And indeed, when we look at the decrease relative to mock and cinnamic acid containing plates, you see that Columbia 0 wild type plants are significantly more sensitive than Chi2. As mentioned, Chi2 is part of a broader signaling module. So we decided to test the other components such as MAX2 and SMAX1 mutants to see if they were also affected similarly in hypocaudal length. And if you look here, the red and the teal lines here on this growth curve, you can see that MAX2 and Chi2 behave largely the same, whereas SMAX1 is more sensitive than the Columbia wild type. And if we take the IC50s of these, we can see that wild type has an IC50 of about 20 micromolar cinnamic acid, Chi2 27, MAX2 33, and SMAX1 13, indicating that the whole module is necessary for normal response to cinnamic acid. Another phenotype of Chi2 is that it has shorter root hairs, generally speaking. And you can see that in this mock experiment, both Chi2 alleles have shorter root hairs than our Columbia 0 wild type. However, when we add cinnamic acid, the root hairs of the Columbia wild type do decrease significantly, but even more so in our Chi2 mutants. Again, if we standardize this and normalize it to the mock treatment, you can see that the reduction in Chi2 is significantly more severe than that of wild type. So in this case, rather than being hyposensitive, we find that Chi2 here is hypersensitive to the effects of cinnamic acid. And of course, as I mentioned, cinnamic acid is the precursor for all anthocyanins. So we wanted to test next whether anthocyanin production was abrogated in Chi2 mutants. And you can see here that after we add cinnamic acid, you get a massive increase in the anthocyanin concentration in wild type, whereas in Chi2 and MAX2, you do not. And in SMAX1, you see an even greater increase or accumulation of anthocyanins. And you can see here in this colorimetric extractions that there's really big differences from the wild type to Chi2, MAX2, and SMAX1 in terms of the amount of anthocyanins you can even see with your own eyes. So given that cinnamic acid clearly has an impact on Chi2-based signaling, we wanted to see if by using the synthetic ligand enantiomeric GR24, could we find more cinnamic acid-related components interacting with Chi2? So in this case, we took Chi2 turbo-ID expressing plants and sprayed them with enantiomeric GR24 or a mock solution. And if we look at this, some of these representative GO terms that were enriched, we find interesting things such as autophagy, osmotic stress response, trichome differentiation. But really most interesting to me is we actually pull out a cinnamic acid biosynthetic process as an enriched GO term. So if we just look first at the mock enriched protein interactors that we pulled out in the proxitome, we can see that SMAX1 is highly enriched in our mock enriched. And this makes sense because when we treat with GR24, we are triggering the degradation of SMAX1. So it is good to see that that we have a positive control showing up in the correct treatment. Now, more interesting was when we looked at the GR24 treated data set, we found that all 4 PAL isoforms in Arabidopsis popped up. And as I mentioned, these phenylalanine ammonium lyses are really interesting, in part because they are the first determinative catalytic step in making the entire phenylpropanoid pathway, and are making cinnamic acid, which we have just seen is very important which requires Chi2 signaling module. We validated indeed that PALs 1 through 4 do interact with Chi2 using, again, a split luciferase system. And you can see here we have MAX2 and SMAX1 positive controls, and that we get the same amount of luminescence for the PALs, indicating indeed we do have really nice complementation and interaction. Given all of this data with the cinnamic acid and Chi2 and that it requires the E3 MAX2, we then next wondered, well, is there any way that cinnamic acid could potentially be a KL? Now, it's not. We tried multiple different things, but what we did find is that when we add cinnamic acid to a ratiometric system containing a SMAX1 reporter using a luciferase fused to a SMAX1 D2 domain, and then a reference M scarlet fluorophore, we actually find that as we increase the concentration of cinnamic acid, we see a degradation of SMAX1 that is similar to that of GR24. Now, mind you, an enantiomeric GR24 is the ligand, or is a synthetic ligand that binds to Chi2, and so at much lower concentrations activates SMAX1 degradation. But we see this interesting trend that leads us to many new interesting questions about what on earth is cinnamic acid doing with Chi2. So we'll leave it there. There are many interesting new things coming up, and we hope to publish a paper that will hopefully explain some of the things we've seen today. But I want just quickly to go through a take-home summary here. So proximity labeling has expanded the Chi2 network and reveals new interactions and new signaling pathways, and indeed does show the power of proximity labeling to help us drive new hypotheses. MLP, a cinnamic acid responsive carrier protein, may promote the stability of Chi2, perhaps in an exchange of molecules mechanism. Chi2 physically interacts with cinnamic acid biosynthetic enzymes, and cinnamic acid physiological responses require Chi2 and impact Chi2 signaling pathways. Okay, all this work, of course, could not have been done in isolation, and I want to make sure I thank all of the people who are involved, including everyone in the Shebeck lab, my boss Nitsan, Malthi, Palyam, who did a lot of our structural work with us, Sunyan, who's been very useful and helpful in doing some of the ratiometric reporting, and then 2 fantastic undergrads that helped us throughout this process. Of course, our collaborators at Iowa State, Justin Wally and Christian. We've been working with Natalia Dudareva at Purdue University and want to thank her and Matthew Bergman for some excellent work that they've done, not shown today, but tangentially. And of course, Caroline Kuchar at Max Planck and Johana Schmidt, her graduate student. Also, thank you to Dave Nelson and Mark Waters for plant material. And thank you all for being here. My pleasure.
Great. Thanks very much. That was great. Brilliant. Right. Okay. After 3 interesting talks on very different aspects of receptors and ligands, it's chance for questions and question session. So please do put your questions in the Q&A box. I can see Satoshi and Johana have answered some of them in writing as well. But we'll take the first question that's still there, which is a ZIP1 question. So the question says, since ZIP1 is activating SA triggered immunity, and SA signaling often involves worky transcription factors binding to W boxes, is this a feedback? Have you looked at whether ZIP1 sort of could actually regulate its own expression? Yeah.
Mm-hmm. Yes, we tested doing some qPCR analysis after SA and CEP1 treatment, if we can get also CEP1 upregulation itself. And we see a CEP1 upregulation with the peptide itself, but also with salicylic acid itself. So, and concerning the W box, so these, we assume this is because salicylic acid and CEP1 works similarly. So, and then after the activation, you get the feedback loop. If this is also timely related is the next question.
So I guess a question for me, because I couldn't type things in the box, I think, because I'm a panelist. What do you see? Maybe I missed it, but the role of this, is it an amplification sort of loop? Yeah.
We do see, so the amplification loop happens through the PLCPs. Yeah, so first in the first paper, we had the idea that the PLCPs might release CEP1. Now we know that the metacaspases are the ones releasing CEP1, and this is timely related. This is what I meant. So ProCEP signaling happens very fast. So basically, we have an upregulation of ProCEP after 6 hours salicylic acid treatment or CEP treatment. It started already at 3 hours, but after 6 hours, it's very high. But PLCP activation happens after 24 hours. So we know then, and the whole salicylic acid signaling, so when we test for PR gene expression, it happens at 24 hours. Yeah, so that means the process from processing to signaling and activation goes over time. Yeah, so, and we know now that CEP1 itself also upregulates ProCEP. So, and then you produce more ProCEP that will then activate PLCPs, and over time then do the feedback for the salicylic acid.
It's sort of got to build up, the ZIP1 has got to build up to a certain sort of threshold before it then activates the PLCPs.
Exactly, yes.
And you propose it's activating the PLCPs directly rather than through transcription, or you
don't think so, but this is we haven't tested this, so we don't see a clear interaction between PLCPs and CEP1, only for cleavage. We actually know that in the apoplas, the PLCPs are cleaving CEP1 itself. So we now believe that actually, because PLCPs are activated later on, this works as a clearance signal, because you don't want to have all the time a CEP1 immunopeptide acting everywhere in the whole immunity pathway. So after the signal or the feedback loop has been done, then the PLCPs are activated and start cleaving then the peptide. Yes, but how exactly this activation happens? Yes,
who knows?
Next story.
Great. Okay, thank you. The next question is about your Turbo ID. So how are you distinguishing between sort of, true Chi-1 signaling partners and the noise, things that happen to be around it?
Yeah, I think this is a pretty insightful question. So, good controls are the most important thing, and then a little common sense. If you are working in something that doesn't exist in the chloroplast, just because and I'm not saying necessarily Chi2 here, but if you know subcellular localization and you find something that's in a compartment that just doesn't make sense for your protein to be in, that might be a good candidate to remove. But frankly, the only way to really know is through extensive validation, and that is, I think, the biggest hurdle in proximity labeling is validating the enormous number of hits. I mean, we get hundreds to almost 1,000 hits depending on which data set you look at. The other thing is, of course, just sometimes using good controls. And if you, for example, use the treatment-based assays might be best in some ways, in part because you have the same level of protein across all genotypes, and that differences in interaction are going to be due to changes in the interaction landscape. Whereas sometimes in using a citrine or whatever, you might end up with a little bit more noise. So that's something to keep in mind, is that good controls and then good validation and verification are the way that we can make sure that we have true interactors versus things that just pop up. And again, I think like proteomics, at some point there's going to come a list of things that appear in every turbo ID data set that you can remove. But we are really, we are maybe 6 years out since Alice Ting first published her paper. So it's not quite, but it's still a relatively new technology.
Great, thank you. That's a really useful answer. And then there's another question about cinnamic acid. Could it be part of, or the sort of, I guess, the ancestors of strygalactone signaling?
So, I mean, we know that strygalactone signaling comes out of the carotenoid biosynthetic pathway. And so we already know that strygalactones are produced in a different pathway than the phenylpropanoids. That said, plant metabolites are extremely diverse. And as I think many of us know, cinnamic acid is a starter molecule for many, many, many small molecules that we use in cancer, small molecules that we use in human medicine, small molecules that plants use for all sorts of crazy things that we don't know. So I think that, it's easy to say might be a starter molecule, but there's a lot more work to be done there before we can definitively say that cinnamic acid is the foundational molecule for a KL type or a strygalactone. And
just a question for me related to that, I was just thinking, so there's no evidence that cinnamic acid binds to MLP?
No, we've been trying extensively to do biochemical verification. We do not find cinnamic acid is binding to MLP in our hands, which is surprising, and yeah, it'd be nicer if it did. But of course, this is something that we're still going through and trying different biochemical approaches. It could be that we're just not using, the right technique to approach this.
Cool. Okay. The next question is another one on ZIP1. When you did your mass spec analysis, you had you just got 4 peptides. So the question is just asking a bit about how did you? I think you said that those were the only 4 you got, but the question's asking how did you select those 4, or how? Yeah, from the data.
Yeah, that's actually a very interesting question, because of course now when we do mass spec analysis, we get a bunch of 10,000 peptides. But at that time, and this is now 15 years ago, the maize genome was not the best, let's say, as for using as a database. And we were very lucky because only in version 3 we could identify these genes. So PROSIP1 doesn't exist in the next versions of maize. So we were very lucky to find this peptide there. And yes, I think it's just the methodology has expanded now. So we have better machines, we have better databases. This is how we find now more peptides. But by the time, it was also the extraction of apoplastic fluid a thing. So we were just starting with all these methods. And we got, of course, more peptides than these 4, but these 4 were the only differentials between the MOC and salicylic acid. And this is how we end up with those peptides, actually. Yeah.
Excellent. Serendipity is good sometimes.
Yes.
Right. And then, there's another question about RLKs. So maybe for Satoshi here, it says, so wondering if you see cleavage of RLKs at the ectodomain. I was saying that they see a group of RLK ectodomains shedding in the apoplast after FLAG22 treatment. I wanted to know if you, I guess all of you, if you see any cleavage of RLKs. I'm not sure if we.
Sorry. Could you repeat the question?
Yeah, I'm not following. So basically, the question says, is wondering if people see the cleavage of RLKs, particularly at the ectodomain. I guess the outside, the outside domain. So basically, this person's saying that they see cleavage of a group of RLKs in the apoplast after FLAG22 treatment. So I suppose whether you see cleavage of the like BRI1 in response to any treatments.
Okay, so now some receptors are known to be cleaved after stimulation, but so far as I know, the BRI1 cleavage is not observed. As far as I know, So yes, is that the answer?
Yeah, no, that's great, Satoshi. Thank you. I had another question. I don't know if you know, but I was thinking about, you obviously got the interaction between Bri1 and your MBAPs. Do you know the stoichiometry of that at all? I mean, is it one-to-one binding, or do you know which bit of the protein is binding? Can there be more MBAPs binding to one Bri1 or the other way around? Do you have any idea about what sort of size complex that makes?
The complex of what? Sorry, sorry,
Sorry.
I cannot.
I'm just saying with your Bri1 binding to your MBAPs, do you know what the stoichiometry of that is? Is it a one receptor to one microtubule associated protein, or yeah?
Yeah, that's a very good question. Nice question. Yeah, that's what I also want to do. So yeah, because Bri1 is only conserved in seed plants, but MBAPs are well conserved in land plants. Yeah, which means MBAPs should have more partners. So yeah, we also wonder such kind of questions. Yes. So yeah, in Arabidopsis, we still don't know, but we are planning to do the term ID as Jakob shows. Yeah. So I hope we can answer more concretely. At some point. Yeah.
Great. Okay. So I think I don't see any more questions in the chat.
Can I also ask a question,
Yes,
Katherine?
go for it. Yeah.
I cannot write in the
No, exactly.
Sorry. So, Jakob, I was wondering if you have maybe tested if your K1 mutants are more susceptible to pathogen infections based
So
on
I have
the one data. Sorry.
Yeah, I have not. I personally have not, but I know that there's there's a paper that recently came out that I should know this off the top of my head, but there's a paper that came out not too long ago that looked at some of the SMAXs, and when you remove them, that you get aberrant defense response. And so I think the connection there was indeed that I think it was SMAX1, and yeah, so I personally have not tried it with Kai2 mutants, but I think that's something that would be fairly straightforward. The big question, of course, is it's another phenotype without necessarily getting at how it works, right? And I think that's one of the big problems with, I think, we're in the receptor field, and one of the problems with receptors is that once you figured out the ligand, which we don't know quite yet for Kai2, then what's interesting is all the downstream mechanisms, right? And so we're trying to figure out where Kai2 sits. And so the hope is that, Kai2 has other post-translational regulatory interactors, or it has other functions that we don't know of yet. But most likely, it's going to probably go through the SMAXs or SMAXels. And then the other thing, not to open up a new can of worms, tricky is to discern some of the differences between D14 and Chi2, so strigolactone versus kerrikin signaling. D14, for example, can target some of Chi2's downstream targets. And so there's some interplay there that's confusing, where it's clear that pathogenicity is affected in during infection, but it's not clear how much Kai2 necessarily is doing outside of the SMAXs, if that makes sense.
Yeah. Maybe just to add something. So have you tested the level of your hormones in the Kai2 mutant? So because I assume, for example, do you say it is more upstream of all hormonal pathways? So if you have a mutant there, then you will have, for example, less salicylic acid.
So yeah, have not done the metabolite studies. Haven't done any hormone quantification there. But generally, looking at the last 20 or so years of data that's available, we are seeing that Kai2 consistently and consistent with its ancient origins is appearing upstream of a lot of our major things. And so, for example, I think the hypocaudal phenotypes that we see in Kai2 is largely thought to be due to downstream pin and auxin regulation, or misregulation rather, in the mutant. So the big question is, does Kai2, how does Kai2 impact, these core hormone pathways, and how does it regulate it? And it's clear that it does, but I think some of the mechanism, again, is lacking unless it's purely described through the SMAX and SMAXL repressionary machinery. Does that answer your question?
Yeah, sure.
Okay.
Thank you.
Yeah.
Great. Well, wonderful timing, everyone, and thank you very much, everybody. So yeah, I want to great thanks to our speakers for sharing their work, and look out for their publications in the Plant Journal, which we'll pull together there. I think at least some of them are already published, and then we'll pull them together into a special issue over the next month or so. Yeah, and thanks to you, the attendees, for coming along, and we hope to see you again at the next webinar. Thank you. Bye-bye.
Thank you. Bye-bye.
Bye-bye, everyone. You will
Bye-bye.
receive the recording, the link to the recording in the follow-up email, and you will receive as well the certificate of attendance, especially the people who are still in the room. Thank you so much for staying until the very end. And see you next month for our next webinar that we will make public soon. And have a great day. Bye-bye, everyone.